The Chris Kine Academy, the YouTube channel which brings you about mathematics, biology, and general science in a worldwide. Now I'm coming up as a Chris Kine. I'm going to teach you about a new video of biology which is all about proteins, RNA, and DNA, what you call ribonucleic acid and deoxyribonucleic acid, the structures and its function, and how it's operates in the living things. So now, as far as I did in last session, I discussed about the first system, and then I'm coming up on the proteins and second number 2. So now we know in usual the varied or the very intricate structures of protein enable them to carry out numerous functions. As usual, the cell so strings together 20 different amino acids. As far as the protein molecule is concerned, we like to find that the protein, to be a molecule, it have to consist almost 20 amino acids in linear chain to form a protein. So the protein commonly range in the length from 10, 100 to the 1000 amino acids. This is almost the quaternary structure of the protein. Like to find there are almost 100 or 1000 amino acids, but some are much shorter and the other are larger. And when we obtain amino acid either by synthesizing them from other molecules or by breaking down proteins that we eat. So the essential or amino acids from dietary standpoints are the 8 that we cannot synthesize and must obtain from food. So it's what we call isoleucine, leucine, resin, methionine, phenylalanine, threonine, tryptophan, and valine. So here we like to find what we call essential amino acids, and then beans and corn together have or either making their combination particularly nutritious. So we find the artificial proteins or amino acid, and then we have the natural amino acids, essential and non-essential. So here we have the hierarchical structure of protein. The protein go through different structure, although constructed by the polymerization of only 20 different amino acids in 2 linear chains, but protein carry out an incredible array of diverse tasks. So the protein chain fold into a unique shape that is stabilized by non-covalent interactions between regions and the linear sequence of amino acids. So this spatial organization of protein, its shape in 3 dimensions, is a key to understanding its function. So only when a protein is in its correct 3-dimensional structure or conformation, it is able to function efficiently. So a key concept in understanding how proteins work is that function is derived from 3-dimensional structure, and 3-dimensional structure is specified by amino acid sequence. So the amino acids proteins are joined by peptide bond. The bond required to be found in between the 2 amino acids, which form it comes from the monomer, and then the many monomers becomes the polymer. Then the polymer, it's all about what you call protein, and then the bond between those many monomers to form a protein, or the bond between those more than 2 polymers is what you call peptide bond, to form a polypeptide chain like nucleic acid. So polypeptide have polarity, with one end having a free amino group, and then other ends possesses a free carboxylic group. Or some proteins consist of only a few amino acids, whereas other may have thousands of these, a few than about 50 amino acids in a polymer. We tend to call it a peptide. So more, and it's a polypeptides. So simply means if we have the structure of amino acid as you see here, we like it to find different parts which consists the protein molecule. Yes. Then amino acid must have the carboxylic group, the amino group, and then the carbon background or carbon backbone. So and then here we have 2 amino acids which combines together to form a polypeptide chain. The polypeptide chain is formed between 2 amino acids, and then in that between those amino acids there is a bond which is called peptide bond. This is the bond required to join together 2 amino acids to form a polypeptide chain, or those polypeptide chains, it's come up to tend to call up a peptide or more than one, and then we call it polypeptide. So as you see here, we have the carboxyl group, which is C double oxygen. Then we have to join with then amino group, and then the hydrogen from amino group, it tends to make to left, and then the carbon, or there is a bond between nitrogen and carbon, nitrogen from the amino group and then carbon from the carboxyl group, and then they form what you call polypeptide bond, and then that molecule tends to be what you call a peptides. And then in addition, we have a water to be provided. So after this, they have, instead of having amino group, they have amino ends with nitrogen NH2, and then inside another side we have carboxylic end, and then after this it is forming a peptide composed of 5 amino acids according to how many monomers we have to be joined together to form a polymer. So here, a polypeptides that folds into a surface shapes are called proteins. If the polypeptides folds into a specific shape, for instance, primary structure, quaternary, or tertiary, it's what we call protein. Here we consider the structure of the proteins at 4 levels of organization. So the starting with their monomic building blocks, the amino acids, we have here a molecular structure starting from primary structure, what you call a sequence. Then secondary structure, it's likely to be a local folding, and then tertiary, it's all about long range folding, and then quaternary, it's a multimeric organization. So amino acid names are abbreviated to same times. We usually write amino acid as either A, 3 letters abbreviation. For example, glycerin is written as GRI, and leucine written as REU, or as a 1-letter code. For example, glycerin is specialized, or it is represented by the capital G, and leucine represented by the capital L. So this is the format we like to use while we are recording amino acids. We like to use 3 letters, or we like to use a 1 letter, which is capital. Then the numeric numbering is always from the amino terminal ND, and then where synthesis of the protein begins on the ribosome. And then if we are arranging this name of the amino acids, we start from the amino group or amino terminal where those ribosome likely to attach. For example, we have rhizosome, and rhizosome, it's an example or is an enzyme that attacks bacterial cell walls. So it's found in secretions such as tears or saliva. So having the following primary structure, this is a structure of rhizosome. We start from the amino group, and then you add there other amino acids, then we end up with the carboxyl group. That's how we construct what we call a protein molecule. So link, we are continuing all about talking about a protein. So like that of nucleic acid, the molecular structure of protein has several levels of organization. So we have, as you see, the primary structure, we have secondary, tertiary, and quaternary. For that one, from primary structure of a protein, it's a sequence of amino acid, just like that, a sequence of amino acid. So through interaction between neighboring amino acids, a polypeptide chain folds and twists into a secondary structure. A real definition of structure, a secondary structure for amino acid, it's the folds and twists into, or the fold and twists of the amino acids. Second structure are formed because of hydrogen bonds of disulfide bonds into, or disulfide bonds. So involved the peptide bonds of the backbone. So two common secondary structure found in protein are the beta pleated sheets and the alpha helix. So the secondary structure interacts and folds further to form a tertiary structure, so which is overall 3-dimensional shape of the protein. So it can be protrusion, clefts or grooves are present on the structure where particularly amino acids are positioned to form sites that binds ligands, and in this case of enzyme, so the catalyst or it catalyzes reactions within a between ligands. So a tertiary structure is unique to particular proteins. And then the secondary structure of a protein are ultimately determined by the primary structure and then the amino acid sequence of that protein. And then here, finally, some proteins consist of 2 or more polypeptide chains, what we call protein subunits, and then that associates to produce a quaternary structure. So this tertiary structure, if it comes on a range of having the subunits protein, this becomes to be associated together to produce what we call a fourth structure, which is called quaternary structure. So these subunits are held together tightly, but not covalently by the same interaction that stabilizes tertiary structure. For example, here we have a hemoglobin. So the protein that carries oxygen in our red blood cells, as usual, so is formed from 4 individual propeptide chains, 2 alpha and 2 beta chains. The three-dimensional arrangement of the protein subunits as they fit together to form the multimolecular structure is called what we call quaternary structure of protein. It's all about the three-dimensional arrangements of protein subunits as usual to fit together, and then if they form what you call multimolecular, it takes a title or a definition of what you call quaternary. As I said, it's almost a quaternary structure consists of 2 alpha chain and 2 beta chain. If formed together, it forms what we call a quaternary structure. Here we have the general structure, which represents primary structure, secondary structure, tertiary structure, and quaternary structure. This is all about first structure. Let me try to use a pen. Yeah, this is all about a primary structure of the protein. Then this is the second structure. If we say the if we said it's amino acid sequence, this is what we call amino acid sequence. Amino acid 1, amino acid 2, amino acid 3, and amino acid 4. Then second structure means the alpha helix and the beta sheets. This is what we call tertiary structure, like an example of insulin, and this is what we call quaternary. And as I said, it's the three-dimensional arrangement of the protein subunits as they fit together to form the multimolecular structure. As you see here, it's a multimolecular structure. And so let's continue now. The function of proteins. There are many, as much as possible, functions of proteins. So proteins are involved in particular or chemical processes in living organisms. As you know, proteins likely to be messengers of different impulses, for instance, in coordination, and then that's likely to be occurred in chemical processes. And those all chemical processes that we likely to find in our body, it's involved by proteins. So their universal significance is apparent in that as enzymes. And they drive chemical reactions in living cells. Though they involve in particular chemical reactions or chemical processes, but also it drives the chemical reactions in living cells. So without enzymatic catalysis, the micromolecules involved would not react spontaneously. All enzymes are the products of one or more genes. Also, again, here we have protein also serve to transport small molecules. It's a protein. You know, there is a process in a biological process likely to be require the protein transfer. So this protein chain, it is used as a transport means of ions or metals. Proteins have important function in cell division during growth and in cell and even in tissue differentiation. So the control of coordination, or they control coordination of movement by regulation muscle cells and the production and transmission of impulses within and between nerve cells. As I said the first time, I told you that proteins are involved in coordination. As here this point says, so the regulating muscle cells, by the time when there is contraction and relaxation of muscles in process of in coordination or contraction of muscle, the protein plays a big role in controlling that coordination of movements. And they control blood homeostasis or blood clotting. So, and the immune defense. So, proteins take a big importance in homeostasis or blood clotting. It plays a big role, and even in immune defense. And then they carry out mechanical function in skin, bone, blood vessels, and other areas. So thank you very much. This is all about proteins and RNA, but this is in a part of only proteins. We see its introduction, what comprises in proteins, what structures are to be found in proteins, and later we find what we call the functions of proteins. So thank you very much for watching this video. I would like to request you to make subscribe if you don't do it, and share this video so that everybody wants to know about proteins can join. Thank you very much. See you.